share my screen. Yeah. Oh, good, that finished, okay. So let me remind you, I want to finish chapter 13, and if possible, get started with chapter 14. I don't know if we'll get that done, but we'll try. This week in the lab, you should be working on your unknown project. It is due Wednesday, August 25th. And that means Monday will be the last day that I will be giving out test requests. So if you have a test you'd like done, make sure you request it before Monday, because after Monday, I will not give out test requests. And I might, if you ask on Tuesday for a test that you can be done in one day, I might let that one slip. But if you ask for a 2-day test on Tuesday, I'll email you back saying that you'll be getting the results on Thursday and the project is due on Wednesday. And so for most of the tests, you have to ask for a Monday before I will send it to you. And then there's one test that takes 5 days, the MR test or the NUFA-RED test. And that, if you ask on Monday, I will not give to you in time. So you need to ask that one. Let me see. Wednesday, so Tuesday, Monday, Sunday, Saturday. You have to ask for an MR test this Friday if you want an MR test. Otherwise, you won't get the results in time. Any questions about any of that? All right, if not, I'm going to close that down. And just pick up where we were left off last time. We talked about the coronaviruses and finished talking about the family of viruses. Let me mention that viruses have an association with cancer. It's believed that some human cancers, 10 to 15% of cancers, are caused by viruses. For example, human papillomavirus, it's well known that it causes or aids in the development of cervical cancer, as well as skin cancer. Hepatitis B virus and hepatitis C virus can lead to liver cancer. However, not all cancers are caused by a virus. For example, breast cancer and glioblastoma are not associated with viruses, at least in humans. Any question about that? So 10 to 15% of human cancer seems to be caused by a virus. Obviously, what is that? 85 to 90% of cancers are not caused by a virus. There are two main ways that viruses can cause cancer. First, you can have the formation of a provirus where the viral genome integrates into our DNA. And that changes the cell's makeup. It can mutate the DNA. So DNA viruses that integrate and retroviruses can cause cancer just by integrating into our DNA and then mutating the DNA. The second way that cancer can be caused by viruses are by oncogenic viruses. These are viruses that carry an oncogene. An oncogene is a mutated form of a normal cellular gene involved in cell growth. So this gene normally allows the cell to grow And obviously it would be turned on in most children because they're growing. But that's the normal gene. And the gene is controlled, and the cell will only grow so much. The oncogene is a mutated form of this normal gene, and it causes the cells to grow uncontrollably. And so when an oncogenic virus infects a cell, it carries this oncogene, which then causes that cell to grow uncontrollably, and that can lead to cancer. Any question about any of that? All right, so the gene can carry a mutated gene that causes the cells to grow uncontrollably. That's one way that viruses can cause cancer. The second way is the formation of a provirus, where the viral DNA is integrating into our DNA, and it can mutate the DNA by doing that. All right. I mentioned that animal viruses can follow the lytic life cycle and the budding life cycle. And then I briefly mentioned there's also a latent life cycle and a persistent infection viral life cycle. The latent viral infections usually happen by a herpes virus, where the virus, when it's causing disease, it's in the budding life cycle, and then the virus goes dormant and enters latency, or the latent life cycle. The virus, when it's in the latent life cycle, remains asymptomatic, and it can be in the cell that way for long periods of time. They do not cause disease, do not cause signs or symptoms while the virus is in the latent life cycle. However, the virus can leave the latent life cycle and then re-enter the budding life cycle. So the virus becomes active. And this is actually what happens when cold sores, a patient gets a cold sore. The first time they get it, the virus is growing, you get the cold sore and is budding and contagious at that point. And that will be caused by herpes simplex virus type 1. And then the cold sore goes away. The virus enters latency. And then later, could be a year later, the next time the patient gets a cold, could be decades later, the virus comes out of latency and then the patient gets a cold sore again. And it doesn't have to be every year. It can be much more frequent than that. The best example of a virus that goes latent is the virus that causes chickenpox and shingles. So a child becomes sick and the virus, while it's in the budding life cycle, the first time it causes the disease that we call chickenpox. And then the virus goes latent, and it can be latent in the patient for decades. And then the virus leaves latency and causes disease. And this time it doesn't ever cause chickenpox. All later times it will cause shingles. And here is when the virus is budding. So it's in the budding life cycle. Any questions about that? The switch between the latent life cycle and the budding life cycle is usually a stress. Like when you get a cold sore, it's because you have a cold. The cold has weakened your immune system, put the body under a stress, and that switches the virus from the latent life cycle to the budding life cycle. Any question about that? Anyone guess what the stress might be for chickenpox? Coming back as shingles? Come on, it's pretty obvious. Who gets chickenpox? Children. Who gets shingles? Usually the elderly. So what's the stress? Come on, take a guess. It's simply old age. The patient is elderly, their immune system weakens, And for some reason, that tends to cause the, or not cause, but be related with the chickenpox virus coming out of latency and then causing shingles. I also mentioned there's a persistent viral life cycle. This is similar to the budding life cycle. It's just it's a naked virus. The virus does not bud through a membrane. What happens is the virus replicates in the cell, most of the time at low numbers, and it leaves the cell by a vesicle. So a vesicle forms around the virus, and then the vesicle will take the virus to the cell membrane. The two will fuse, and then it'll dump the virus outside the cell. Persistent viral infection can cause disease. These diseases occur over a long period of time. The detectable virus infection, meaning the detectable levels of the virus increase over a long period of time. The virus usually is never cleared by the host immune system. In some cases, it may be fatal. In the early days of HIV and AIDS, HIV followed the persistent viral infection, and that is when the patient got AIDS, they got more and more load of the virus, more and more virus being made, and the patient just got sicker and sicker and then eventually died. Subacute sclerosing panencephalitis is another example. It's caused by the measles virus. You don't need to know that, I'm just mentioning it. It can be fatal in patients. Many of the persistent viral infections are not fatal, and they may be tolerated by the patient. Like cytomegalovirus is an example. Varicella zoster, meaning the chickenpox virus causing shingles. In some patients, like my mother, she got the shingles and they never went away. She had them for over 3 years before she died, and she never got rid of them. So it was a persistent viral infection, and it was just tolerated by her system. She was continuously shedding varicella zoster and was continuously giving her shingles. There are a few other herpes viruses where they can be tolerated and they're constantly being shed from the patient. Normally, when a patient gets a viral infection like the cold and the flu, you get the virus and then it quickly rises in the number of virions. The patient gets sickest in this point up here. And then the virus goes away. The immune system mounts an immune response, killing off the virus. And then the virus is gone from the patient. So very quickly, the patient gets infected. Very quickly, a very high virus load. The patient gets sick, and then the virus goes away, oftentimes because of the immune system. Any questions about that? It's called an acute infection, acute and viral infection. A latent infection, this would be an acute infection. The child gets chickenpox, and then the virus goes away, goes latent, and there's no signs or symptoms. So we have no virus being shed from the patient. And then years later, maybe in old age, you can get the latent infection, meaning the virus comes out of latency and starts budding again. You'll notice that in that case, the virus is never as high as the first case. But usually with the latent infection, it usually goes away. And then the patient's shedding 0 viruses again. And during this time is when the virus is latent. In this point, the virus is budding, and this point, the virus is budding. We call that a latent infection because it's after latency. A persistent viral infection is when the patient becomes infected. They have virus at low levels for a long time, and the virus load just accumulates with time. And then it blooms. And you notice how it's broken here? It's because the patient died. This would be the shedding of HIV in the early days of AIDS. And this would be when the patient comes down with signs and symptoms of AIDS, and then the patient dies. So a persistent infection. Panencephalitis, I didn't say that. Subacute sclerosing panencephalitis would be another example of that when it causes death. It doesn't have to cause death. For example, nowadays with HIV, because of the treatment, the patients are living a long life. It might be slightly shorter than a normal human lifespan, but it's close to a normal human lifespan. So they're living a long time. Now, obviously they're under medication, but the point is the patient is going like this, and some of them never develop AIDS. So it just goes like that until the patient eventually dies of old age. But the point is it's a persistent viral infection, and those infections, they just tend to get worse with time. And the longer you have it, generally the worse it is. You can get better. An example is persistent viral infection causing mononucleosis. It builds up with time. This is when the patient gets mono, has signs and symptoms of mono, and then they get better. And eventually it goes away. Any question about any of that? All right, if not, let's talk about prions. That's it for viruses. Prions are proteinaceous infectious particles. This is a disease caused by an infectious protein. It can be inherited and transmissible by ingestion, transplant, and surgical instruments. And in the cases where it's inherited, there is a genetic makeup where the patient is predisposed to getting or forming prions. We'll talk about that in a minute. And so it runs in families. Usually when it's inherited, the patient doesn't develop the prion disease while they're a child, or even when they're a young adult. It usually comes up in middle age. And so the patient may not even know that they are carrying this genetic makeup, although it runs in their family, so they should know. And the point is they have the children, and then they start developing the prion disease after they've already had children. Prions cause spongiform encephalopathies. This is vacuoles in the brain. It sort of looks like a sponge, and I'll show you a picture of that. Well, I'm going to talking about it. I'm going to show you now. That's why it is the name spongiform. There we go. There's a normal human brain. Don't ask me who volunteered to give that sample. It might be a patient that died. And this is a prion infected brain caused by Kuru, classic Creutzfeldt-Jakob disease, also a prion disease causing less plaques than the Kuru, but they still have plaques. And then scabies, this is probably in a sheep, showing the plaques in the brain of the sheep. In animals, the most common example is bovine spongiform encephalopathy, which has a more common name called mad cow disease. But it also causes scabies in sheep, or cervid chronic wasting disease, cervids. Scabies is a disease that sheep, not farmers, what do they call those? Ranchers have been dealing with in centuries in Europe. The sheep would come down with scabies, and the rancher would try and get rid of it, and it kept spreading. So it does spread from sheep to sheep, and I'm not sure they ever figured out how it spreads, but it definitely does spread. In humans, we don't call it that, even mad cow disease, when the British came down with that. They call it Creutzfeldt-Jakob disease. And it can also be called Gerstmann-Straussler-Schechner syndrome. This was a syndrome, and same with Creutzfeldt-Jakob disease. Families that came down with the prion disease, and they found it in certain families. I think Creutzfeldt-Jakob were the two people who discovered it. I don't think those were the two families it was running with. And the same with Straussler-Schechner. They're the two people who discovered it running in certain families. And like I said, these are families that have a genetic predisposition to getting prion disease. We'll explain that in a minute. Fatal familial insomnia, and then Kuru are all cases of prion disease in people. How, I was going to talk about that. I guess I am. Let's talk about Kuru. This was a disease found in the Fore tribe in Papua New Guinea. Let me draw that up. Papua New Guinea over here, the Fore tribe in red. It was a disease found in the Fore tribe because the Fore tribe had a very unusual funeral ritual where they cooked the dead person and then consumed their dead relatives. And that led to Kuru spreading among the Fore tribe. Probably what happened is a long time ago, somebody came down with a prion disease and the Fore tribe had this ritual, and then they ate the person, and especially with the nerve tissue, which is more than the brain, but the brain would be the worst. You then consume the prion, and then the person who consumed the prion would get prion disease, inability to walk, loss of the ability to swallow or chew, leading to weight loss, and eventually leads to death. Any questions about that? Already talked about that slide. What's happening with the prion disease is there are two proteins. One is a normal protein made by a normal nerve cell. It's called the PRP, what do they call that, sub-C or whatever, C. The normal cell protein is involved in normal functioning of a nerve cell. So the nerve cell needs this protein. And then there's a PRP, what do you call that? Hyperscript SC, SC for Scrapies. And this is the prion form of the protein. These two proteins have the same sequence of amino acids. The difference is the folding pattern of the PRPC protein is different from the folding pattern of the PRP hyperscript SC protein. So the same amino acid sequence, the protein just folded into a different pattern. And the PRP SC protein, how it replicates is it comes up to the normal protein, the PRPC protein, and then changes the folding pattern of the PRPC protein so that it folds into the PRP Scrapies protein. And now the cell has two Scrapies proteins, the original one and then this new one. And then the brain cell or the nerve cell says, oops, I need more PRPC protein. So it makes more of it. The trouble is this protein can come up against this protein. There's now two of them. So it's going to happen more often. And once that happens, the normal protein will be converted into the Scrapies protein. More and more Scrapies protein accumulates in the nerve cell or the brain cell, forming the plaques in the brain cell, and that leads to cell death and then the prion disease. Any questions about any of that? So in the inherited cases of where Scrapes disease or prion disease runs in families, what happens is they have a genetic makeup for this gene, which for whatever reason, the genetic makeup of this gene is more likely to convert to this gene on its own. And then once the first does convert, it can convert the other normal forms of the protein to the Scrapes protein. Any questions about any of that? All right, so prion disease, it mostly hits, other than the families that it runs in, it mostly hit Great Britain or England, whatever you want to call it, during the mad cow epidemic. And that was caused because the British wanted to do something with cow brains. Nobody wanted to buy them. Nobody wanted to eat them. And so what they did was they took the cow brains, ground them up, and then added it to grain, and then fed that grain to other cows. Well, one cow came down with prion disease. They took the brain of that cow, put it onto the grain, fed the grain to other cows, and pretty soon we had an epidemic of mad cow disease in England. Any questions about that? And then because people were eating cows as well as cats, the cats and the people came down with prion disease. I think around 150 British citizens and 150 British cats came down with prion disease. Surprisingly, dogs did not come down with prion disease in this time period, so they must be a little more resistant coming down with prion disease than people or cats. Any questions about prions or prion disease? If not, let's briefly talk about plant viruses and viroids. Plant viruses resemble animal viruses in many ways. Plant viruses cause many diseases that are economically important, like important crops like beans, corn, sugarcane, and potatoes. The only one I'm going to mention is tobacco mosaic virus, which can cause disease in tobacco plants, as well as in tomato plants and potatoes. And if the plant gets this virus, they will be much less likely to produce a large bumper crop. So they'll produce a small crop. So instead of having a lot of potatoes, you'll have a few potatoes, we'll word it that way. And the same with tomatoes and tobacco. Because of the tough cell wall, plant viruses usually enter through wounds on the plant, such as by a parasite, like a nematode or an insect, or maybe some fungal disease wounds the plant, and that allows the virus to get in. And then infected plants tend to spread the virus either in pollen or in seeds. Any question about plant viruses? That's all we're going to talk about plant viruses because they don't cause disease in humans. Let me talk about one last thing, viroids. Viroids are naked RNA molecules that cause disease. So you can think of them as a virus without the protein coat. An RNA virus, it's a naked virus. Now remove the protein coat and all you have is the naked RNA molecule. This RNA molecule causes disease. Fortunately, viroids are only known in plants. There are no known viroids that cause disease in animals. The example, I don't have a picture of it anymore, was an infectious RNA molecule that causes potato spindle tuber viroid, which caused smaller potatoes. So the farmer, instead of getting nice big potatoes, they get little potatoes if they're infected with this viroid. We won't talk much about viroids because they don't cause disease in humans. And the last thing I want to mention, recently in the last 10 or 20 years, we've found the largest viruses that for some reason infect amoebas. The very largest virus is Pandora virus, which is approaching the size, the length definitely, of a bacteria cell. And then even the diameter, that's a little larger than the smallest bacteria cell. Any questions? All right. If not, that's it for viruses. Let me just start lesson 14. The reason why I want to get started on it is because I don't want to be spending a lot of time during finals week on covering new material. Chapter 14, Principles of Disease and Epidemiology. Know the terms. I've only got 3 here, but if you learn the terms of this chapter, you'll learn 50% of the material. So this chapter is largely terms. Pathology, etiology, microbial antagonism, know what those are. Understand the relationship between the normal microbiota and its host. Understand the stages of disease development. And then lastly, understand what nosocomial infections are and how to control them. This is one of the most important topics that you can learn about in microbiology, because this is the disease a patient gets from staying in a healthcare facility. So this is also called healthcare facility disease, something like that. Hospital-acquired infection. That's what it's called, hospital-acquired infection. So when a patient stays there, they can come down with a disease that they would not get unless they were in a healthcare facility. Know the terms: pathology, the study of disease; etiology, the study of the cause of disease. For example, the etiological agent of the disease anthrax is the organism Bacillus anthracis. Pathogenesis, the development of a disease. Infection, colonization of the body by pathogens or the presence of a microbe where it's not normally found. For example, E. coli is normally found in the intestines, but if it's found elsewhere, like the ear infection or in the bladder, that would be an infection by E. coli. A disease, an abnormal state in which the body is not functioning normally. We should come to understand that few microorganisms are pathogenic. Few microorganisms cause disease. Some even benefit the host. And that would be especially the bacteria of the normal microbiota. They benefit the host. The normal microbiota is a large community of microbes that are found in association with all animals and plants. These communities are known as the normal microbiota. So when we're talking about human normal microbiota, you should say human normal microbiota. Most people just shorten it to the normal microbiota. These are microbes that permanently colonize a host. So your normal microbiota, once they've colonized you, they will stay with you generally for your lifetime. They're generally found only in certain locations, certain regions of the body. And then disruption of the normal microbiota host balance appears to be associated with diseases and impaired immune function. You have your immune function that's impaired by taking like immunosuppressive drugs, for example, and you can disrupt the normal microbiota, which can then lead to disease. A classic case in point is when the patient takes antibiotics that disrupts their normal microbiota, that it kills off a lot of their microbiota, and then that can lead to a patient's disease, like the yeast infection can come after a patient takes antibiotics. The normal microbiota is normally found on and in the human body, all surfaces, mainly the skin, but also in the eyes, the mouth, the upper respiratory system, the gastrointestinal tract, the lower urinary and reproductive systems, meaning the urethra, are normally colonized by the human microbiota. The lungs are not usually colonized by the normal microbiota. Examples of places where the normal microbiota are not found includes internal tissues and organs like the blood, the cerebrospinal fluid, muscle, nervous tissue, bone, all of your internal organs that are closed to the outside, like your heart, for example. A transient microbiota is different from the human microbiota in that they may be present for days, weeks, or months. But unlike the normal microbiota, they're not going to be with the patient for approximately the patient's lifespan. An example of a transient microbiota that I had was I went to visit my aunt, and she had a Staphylococcus aureus infection. And she, even though she was being treated for Staphylococcus aureus, insisted on hugging me. Okay. The next time, which I think was the same week I visited her, I, at that time, tested my skin for Staphylococcus aureus. It's just something I did about every lab. I would have the students test their own skin to see if they were a patient who had Staphylococcus aureus. And for years before that event, meaning visiting my aunt, I never had Staphylococcus aureus. And then the one time after she had it and she hugged me, I had Staphylococcus aureus. Fortunately, it was a transient microbiota because during the next term, when I sampled my skin, I no longer had Staphylococcus aureus on my skin. So that was a case where, at least for me, Staphylococcus aureus was a transient microbiota. And I'm sure I picked it up from my aunt because she was being treated for it. She had a skin infection from Staphylococcus aureus. Any question about any of this? If not, I think I'll stop here and we will continue with this point next time. Any questions? All right. I will see you in the lecture next time. Come on.
Thank you.
You're welcome. There we go.